All right, for fertilization, remember what we have joining forces here is our oocyte, right? It won't actually become an ovum until fertilization is complete. We'll see how that happens. This oocyte, it was actually a secondary oocyte, that was released from the ovary during ovulation. Now, this oocyte is actually paused in metaphase of meiosis II. I'll just say metaphase II, right? So you can actually see there the chromosomes being lined up along that metaphase plate, right? So we have not completed meiosis II yet, but we are at this point a haploid cell, right? So we have 23 chromosomes here in the oocyte rather than 46, right? Rather than 23 pairs. But that's not the only thing that is carried by the oocyte, right? During the development of that follicle, and as our oocyte is progressing through its developmental stages, it's going ahead and accepting a lot of materials from that follicle, right? So we see things getting packed in here. They'll call them inclusions, right? Often that's things like different nutrients. We'll actually see messenger RNA strands being handed off to the oocyte, lots of proteins, enzymes, things like that, right? So all the organelles that are going to become part of our new organism are also here in the oocyte. Recall that our sperm, right? Our sperm are basically just bringing the DNA. They've totally completed meiosis. They also have 23 chromosomes, right, and are haploid, right, half the genetic material. When this sperm and egg then join, we'll go through the process in more depth, but fertilization, when that takes place, our end product is going to be this single cell called a zygote. That zygote then has our 46 chromosomes, right? It's returned to that diploid state, right, that we would hope to find in the next round of human life. So let's take a look kind of at how this takes place, okay? So this is figure 29.1b that kind of walks us through fertilization. So here we see our secondary oocyte paused in metaphase of meiosis II, right? And they're also showing us the zona pellucida. Recall this zona pellucida is glycoprotein. We also have the corona radiata. These are actually follicle cells that left the ovary with that oocyte. And they're actually, both of these layers are serving as some protection here, right? So now we see the sperm meeting up with the oocyte. The sperm have had to travel a super long distance to get here. And recall that in order for them to even be able to swim, they needed to complete a process called capacitation. When the sperm were just in the seminiferous tubules and even in the epididymis continuing to mature, they were completely immobile, right? It was actually when they came in contact with the seminal fluids that they started to be able to swim, and then they also need the conditions found in the female reproductive tract to complete this process of capacitation, where basically they're able to swim, and the acrosome, right, the tip of the sperm where all those enzymes are, is also able to function. So if we want fertilization to occur, there is only going to be a single sperm that fertilizes the egg. However, you actually need lots of sperm to make this happen. So here's what it looks like. In each ejaculation, there's approximately 200 million sperm. It's kind of a crazy number to imagine, right? But of that, only about 10,000 make it up into the uterine tubes, and then only, I shouldn't say, not a handful, but a few hundred are going to make it onto the isthmus, which is actually the most common place that fertilization is going to take place, right? It takes dozens of sperm to actually break through the corona radiata, right? They all are going to be using all these sperm here to get through the corona radiata. They use the hyaluronidase from their acrosome. Hyaluronidase, right, that enzyme to start breaking through the corona radiata, right? And one single sperm then is going to come in contact, I guess it was this guy, on a receptor on the zona pellucida, right? So a single sperm binds to the sperm receptor on the zona pellucida. Right? So while it is a single sperm that completes fertilization, we actually need dozens, right, to make this happen. In fact, a male who has a sperm count of less than 20 million sperm per ejaculate is considered functionally sterile. Okay, now once we bind with this sperm receptor, this is going to set off a whole series of events in the oocyte. So the one lucky sperm that actually makes it to this receptor, the acrosome is going to rupture, and that sperm's hyaluronidase and another enzyme called acrosin are basically going to digest through the zona pellucida. Right? So the acrosome ruptures hyaluronidase and acrosin. Both are enzymes that make it through the zona pellucida. They basically digest the zona pellucida. Okay. Once through the zona pellucida, we'll actually see, and I don't think this picture does a good job, but we'll actually see the membranes of the sperm and the oocyte fuse. Okay, when those membranes fuse, we get this whole process called oocyte activation. And in oocyte activation, we have three main things that take place. The first of those is what's called, let's say this. In oocyte activation, when those membranes fuse, maybe I'll step back. The membranes fuse, and that increases the permeability to sodium. Sodium starts rushing into the oocyte, right? It's depolarizing. And that depolarization causes an increased level of calcium from the endoplasmic reticulum to dump into the cytoplasm. To me, this is kind of interesting. It sounds an awful lot like the way the nervous system worked, and the muscular system. Sodium was causing the depolarization, and then in the muscular system, from that sarcoplasmic reticulum, which is really just endoplasmic reticulum, the calcium rushes in and allows cross-bridging. But anyway, if that helps you remember it, this increased level of calcium then, we'll go there, is what kind of kicks off oocyte activation. And the three parts of oocyte activation then are what's called the cortical reaction. And basically what's going to happen here is that zona pellucida hardens. We basically make it so that no other sperm can bind. So we say this prevents polyspermy. Polyspermy. So if the egg has 23 chromosomes and the sperm has 23 to get to 46, if another sperm is able to donate its DNA, now we're up to 60, I've lost track, 69 chromosomes, you're no longer human, right? So we want to prevent that. So we prevent polyspermy through this cortical reaction. Another thing that takes place as part of oocyte activation is that the oocyte actually completes meiosis too. Finally. Right? And so that secondary oocyte goes ahead and becomes a true ovum. So remember, if fertilization doesn't occur, if a sperm never makes it to the oocyte, it was never even an egg, right? It was never even a true ovum. And then the third thing that happens is a really rapid increase in the metabolic rate of that cell, right? So at this point, I suppose we can call it a zygote, but if you see an increased rate of metabolism in the oocyte, that's what we're talking about. So some of those things like mRNAs that the oocyte was carrying, now those start to get used. We crank up protein production and all those things as part of this oocyte activation. So once that's completed, I know I'm still just on the first box here, but once all that's completed, then what we see happen is the formation of what they're calling pronuclei. That's probably off the board. Right? So the pronucleus, I think they're just kind of indicating like it's a precursor to the true nucleus. Maybe it doesn't have that nuclear envelope, right? So maybe it's not quite a true nucleus. But what we're going to need here, right? Again, 23 chromosomes here, 23 chromosomes here. Think about this. In the male pronucleus, right, here's, say, chromosome 1, right, and here's chromosome 1 in the female pronucleus. What we need to have happen at this point is DNA synthesis, right? They actually each need to build the sister chromatid, so they look like normal chromosomes again. And then we're going to get what's called amphimixis. They're going to go ahead, these two pronuclei come together, and just like we would expect, there's the chromosomes get close to each other. We'll put it that way. So you have pronuclei development, you have DNA synthesis, and you have what they call amphimixis, right? We're going to bring those two pronuclei together, right? So they mix together, they join. From there, we're just going to start undergoing mitosis, right? At this point, right, we have now a zygote, a new human, right? That zygote, that fertilized cell. From there, we're going to start an awful lot of division taking place, but what you'll notice is they call it cleavage, right? It is mitosis, we're getting mitotic division, but what's going to take place here is no intervening cell growth. So normally when we talk about mitosis, right, if you took a cell and split it in half, the daughter cells would be smaller, they would regrow before they divide again. But in cleavage, it's just division after division after division, right? Oh, sorry, the figure goes this way, right? So here we actually see the pronuclei, that amphimixis, when the DNA all comes together as one happy zygote, and then cleavage is going to begin.